And now picking up in the second order moment here of the cell biology of the nervous system is we actually are looking at how a Schwann cell forms in the peripheral nervous system. And you can see here that we have the Schwann cell, and it's actually wrapping itself around. You can see that there's a central nucleus in cytoplasm. Right here is the nucleus, and the cytoplasm is wrapping itself around, and then it wraps itself more and more times, squeezing down so that there's many layers of membrane ultimately wrapping around. And then finally, here's the photomicrograph of that situation. Okay. Then we're looking at various kinds of neurons. Here's the multipolar neuron that has all of this dendrite incoming information here. You can see that that's the receptive region here, according to the little information bar. Okay, then we have a conducting region, and then we have the region that's going to be what's referred to as secretory. That is to say that the neurotransmitters will be transited down to this location for crossover at the synapse. Okay, so the trigger zone is in this region through here. Technically, there is a trigger zone down here, but it's the opposite of trigger in the case of certain, we have opiates in our own body, endorphins and enkephalins, that can actually act here to inhibit the event. Okay, so here we have a bipolar neuron, which is just conducting. It's sort of like crossing a boundary. In the nasal cavity, we have this with the with cranial nerve number 1, the olfactory nerve. In the retina, in the cranial nerve 2, with the optic nerve, we need these connecting types of cells that connect the receptor to the main cranial nerve. Okay, and then over here, we're having, this is a very elongated receptor where we're getting triggered here, and the incoming information is passing ultimately into the spinal cord. Okay, so generally speaking, when we're looking at incoming information, that's a dendrite. When we're looking at outgoing information, that's an axon. That's just the convention that we utilize. And I'm going to take a look at this event right here, only I'm going to have it as a collection of information right here. Okay, so I've drawn you a membrane. This is what I would normally do in the classroom. And so here are the phosphate heads, the fatty acid tails. This is basically just like a triglyceride with a glycerol and 3 fatty acid tails, except here we've taken off one of the fatty acid tails, turned it around, and replaced it with a phosphate head. But we still have a glycerol right here, and now there's only 2 fatty acid tails. Okay, so this is the nonpolar portion, and then this is the polar. This is the part that is able to be dissolved in the watery substance of our tissues. All right, so membranes can work properly in the tissue situation, and all cells are surrounded by this unit membrane. Okay, and there's going to be things that live in the membrane, transmembrane proteins. Here we have a pump. This pump is pumping sodium out and potassium in. It uses ATP to affect that result. Okay, this is the outside, out this way, the outside. This is the inside of the membrane. Alrighty, and we're going to come down the membrane here a little bit and notice that when we pump the sodium out, we have a high concentration of sodium outside of every cell. Generally speaking, all fluids outside of cells, whether it's blood or interstitial fluid, are rich with sodium because this is happening in every cell in the body. Every cell of the body is actually pumping potassium in. So intracellular fluid is always rich in potassium. That's just the way it is. Here in the membrane of the neuron, at rest, the sodium channel is closed. This is a sodium channel. It's closed at the moment. Okay, so it's bouncing off and we're retaining our sodium out here on the outside. Then we have a potassium channel. Basically, this exchange here happens with sodium and potassium. One positive ion out, one positive ion in. So we have rich interior cellular fluid with potassium. And the potassium channel is closed. In this case, however, there is a bit of leakage. And so it's closed but leaky. A little bit of potassium does flow out and contributes to the positive charge on the outside. And that means that we have a fall of parity. In this case, we have an increased charge, a slightly polar situation on the outside, an increased positive charge on the outside. This is called the resting potential, and it can be measured as minus 70 millivolts. So as we come back to the text here, and we see that we have our little potentiometer here, as it's called, or voltmeter, and sure enough, we have measured in this little cartoon here minus 70 millivolts, just the way you like it. Okay, so that leads us over here to resting potential. Notice we're at 0 here. Here's the minus 70. If it was the heart and we're also dealing with calcium as part of this program, we would be down here at minus 90. But we're not. We're just dealing with two simple ions here, sodium and potassium. This right here is referred to as the threshold. They're not pointing that out. But basically, whenever we have a receptor that is activated, whether it's a light receptor in the eyes or a touch receptor at our fingertips or a sound receptor in the hearing mechanisms, then we're going to get a little bit of a depolarization here, excitatory moment. This is called an EPSP, excitatory postsynaptic potential. Okay, you can check that out, EPSP. If it's significant enough to reach threshold, we'll actually get an action potential. Over here, we're noting that there is an IPSP, which is an inhibitory event. In this case, we're allowing the potassium to flow. In this case over here, we're allowing the sodium to flow. And you can see from our diagram that if I open the potassium channel, the positive charge on the outside will flow in and will join a negative charge on the inside. That's depolarizing. Right, so we're polar at this moment in the resting potential because there's more positive outside and more negative on the inside. So if I open the sodium channel, I will depolarize. If I open the potassium channel, I will hyperpolarize, okay, or inhibit. Okay, and then we're moving over here to the action potential. The action potential here is showing us that this event is propagated by the flow of sodium. So increased sodium permeability results in depolarization. It's an all or nothing event once we get past threshold. All right, with me? Okay, so we're getting past threshold. We're actually crossing 0 into this, the far reaches here of the peak of our action potential. The sodium channels will close at this point, and we're going to now get, see it's closing, and it's getting pumped back out again. Here the potassium channels have opened. It's a slower channel, and it's repolarizing. Here we have the repolarizing event, where we're going to get the sodium back out, and we're going to pump the potassium back in. Okay, and so ultimately we get back to the resting potential. That was a little quick, okay, but I have to pause here and start another little event.